Hello guys, can you hear me and can you see the presentation? Okay, hello. So I will start. So today we discuss nervous tissue. Nervous tissue consists of cells, neuron cells, the major type of cells, and glial cells or just glia, accessory cells, cells that help neurons to perform their function. As you know, tissues consist of cells and extracellular matrix, but in nervous tissue, like in epithelial tissue, we have almost no extracellular matrix, and cells are located very close to each other. In this picture, you can see the large ones. These are neurons, the major cell type, and only cell nuclei of glial cells. Neurons are the cells of static cell population. It means that they cannot divide anymore in adult organism, and we have almost no stem cells for neurons. But glial cells are the cells of growing or stable cell population. That means that they can proliferate and then can be regenerated by division of cells. Histogenesis of nerve tissue starts when neural tube starts to be formed. This process starts at day 18 of embryonic development, and this process is called neurulation. First, neural plate forms, and then it becomes to be closed, and the closure begins at day 21, and its final day is about 25. And some cells are not included, not into ectoderm nor into neural tube, and there they form neural crest. And also in the frontal part of the embryo, ectoderm makes the thicker areas, which are called neuroectodermal placodes, and they also give rise to some nerve tissues. So we have three sources of development of nervous tissue. Neural tube, it gives neurons and glia of central nervous system. Neural crest gives rise to glia and neurons in peripheral nervous system, which includes autonomic and sensory ganglia, sensory cells, and also pia mater and arachnoid mater of brain. These are coverings of brain and spinal cord. Ectodermal placodes give rise to ganglia of cranial nerves 3, 7, 9, and 10. Here you can see the processes which take part in the developing neural tube. This is the neural tube. It has three zones, three areas. The innermost zone, the layer of cells which surround the cavity, which is called neurocell. This layer of cells is called ependymal layer. It consists of progenitor stem cells. Here they divide. They divide, proliferate, and their daughter cells migrate into second zone. Yellow in this picture. It is called mental layer. Here the cells which are called neuroblasts and glioblasts start to develop into neurons and glial cells respectively. So in mental zone we have maturation of neurons and glial cells. While maturing, the neurons grow their single axon, the process, and this axon grows to the periphery, and the axons of maturing neurons form the outermost marginal layer. So marginal layer, the outermost layer of developing neural tube, it consists of neuronal axons. Also in this picture you can see the developing spinal ganglion or the dorsal root ganglion, which is the sensory ganglion. It consists of groups of sensory pseudo-unipolar neurons. So spinal ganglion develops from neural crest. Here previously was the neural crest, and all the neurons and glia outside of the spinal cord and brain develop from neural crest. So here you can see the processes which take part in neural tube. Once again, in ependymal layer we have proliferation of stem cells. In mental layer we have migration and differentiation of the cells. In marginal layer we have the axons of neurons and those neurons which didn't find their target, the cell or tissue which should be innervated with the axon. Those neurons die with apoptosis. So only those neurons develop which find their target cell, which is innervated by these neurons. We have four types of neurons according their structure in histology. So this is the morphological classification based upon the structure of neurons. So, unipolar neurons have cell body or the soma and one single process axon. Only one axon, so it is called unipolar. This type of neurons is present only during embryogenesis in neural tube. Next, second one, bipolar neuron. It has one axon and one dendrite, which grow from opposite sides of the neurons. These neurons are afferent, it means they are sensory, they receive sensory information, and they are found in retina of the eye, which serves to receive the signals about the light, in spiral ganglion of the inner ear. So there they receive information about the sound. Third one type, pseudo-unipolar neuron. Pseudo means looks like, but not really is. So pseudo-unipolar looks like unipolar, because like in unipolar neuron, one single process grows from the cell body, but soon after it splits into two processes, one dendrite and one axon. So actually, pseudo-unipolar neuron has two processes, one axon and one dendrite. And these neurons are found in dorsal root ganglia, which are sensory ganglia. And finally, multipolar neurons. They have many dendrites and usually one single axon, which can branch at the periphery. So one axon, many dendrites. Most of all neurons in our body are multipolar neurons, and all the neurons in brain and spinal cord are multipolar neurons. We have other classification of neurons, for example, classification which is based upon the position of neuron in the reflex arc. So according to this classification, we will divide neurons into afferent or sensory, interneurons, and efferent or effector motor neurons. Also, we have functional classification, which is based upon uh, how neurons act upon other neurons, such as inhibitory and excitatory neurons, but now we discuss only histological classification, which is based upon the structure of neurons. So, here you can see the structure of the multipolar neuron. It has many dendrites, which are branchy processes. They grow from the cell body, which is called also cell soma. This is Greek for body, soma. Uh, and from this soma grows one axon, which is branchy at the periphery. A cell body or the soma includes nucleus with well-developed nucleolus inside it, and the surrounding cytoplasm, which is called perikaryon. Karyon stands for nucleus in Greek, and peri means surrounding. So this area is also called perikaryon of a neuron. Neurons are static cell populations, so they cannot divide anymore. The major function of neurons and the major function of nervous tissue is to generate and provide nerve impulses. Nerve impulse runs from the periphery of dendrites towards the cell body, then along the membrane of the cell body, and after all, along the axon from the body to the periphery. And these arrows indicate the direction of nerve impulse. So this is very important. Nerve impulses never go in opposite direction, only from the tips of dendrites towards the cell body, and only from the cell body through axon to the axon endings. And this is very important to understand the structure and function of neurons, the nerve impulse. Nerve impulse will be discussed in physiology course, but now we will uh, talk talk about it not in details but in general. So, nerve impulse, or it is also called action potential, is a wave of depolarization of the plasma membrane of a neuron. It runs along the membrane from the ends of dendrites towards the cell body, and then from the cell body through the membrane of the axon to the end of the axon terminals. So, nerve impulse is the wave of depolarization. What means depolarization? It means that in resting state, the membrane of a neuron is polarized. It means that with the help of sodium-potassium ion pumps, which are transmembrane proteins incorporated into bilipid layer of the membrane, with the help of energy of ATP, the sodium ions are pumped out from the cell and thus creating positive charge outside of the membrane and negative charge inside the cell, the inner side of the membrane. And this is called the resting potential. It means that normally in resting state, the membrane of a neuron is polarized. And then, if the neuron receives some irritation, which is the signal, usually signal from other neuron with the help of neurotransmitters, this irritation causes the opening of um, sodium channels. Sodium channel is the transmembrane protein, also incorporated into bilipid layer of the membrane, but it doesn't use the energy of ATP, and it allows ions to pass through it passively from the highest to the lowest concentration along the concentration gradient. In resting state, these channels are closed, so sodium ions cannot pass backwards from outside to inside of the cell. But when irritation happens, these channels open and sodium ions go very fast, very quickly into the cell. And this causes change of the charge of the membrane in this particular area from positive outside and negative inside to the opposite, negative outside, positive inside. And this causes the nearby uh, ion channels also to be opened. And this process happens in this area, then in this area, and then in this area. And so this causes the wave of depolarization of the membrane, which runs along the membrane like the wave. So nerve impulse is the um, action potential, the wave of depolarization, uh, which is provided by the presence of very high amount of ion pumps, sodium potassium pumps, and ion channels incorporated inside the membrane of the nerve cell. So here you can see how the wave of depolarization runs along the membrane of the axon from the cell body towards the periphery of the axon. So, neuron structure, ultrastructure, which means the inner structure of the cell, with its organelles includes the cell nucleus, which is usually very large and light because of prevalence of euchromatin, which is the active form of DNA. Um, in neurons, we have a lot of euchromatin because neurons provide production of very high amount of proteins, mainly those proteins which are incorporated in the membrane, which are ion channels and ion pumps. So these are the proteins which should be produced inside the cell. So we have large and light nucleus with nucleolus. As you remember, the function of nucleolus is to form ribosomes. Ribosomes are needed to provide the synthesis of proteins. Next, we have a lot of Golgi apparatus and a lot of rough endoplasmic reticulum. So, rough endoplasmic reticulum, polyribosomes and ribosomes together form the patches which are called Nissl bodies. So, neurons have very high amount of protein synthesizing apparatus, which includes rough endoplasmic reticulum, polysomes, and Golgi apparatus, to make a lot of proteins needed for impulse transmission. Next, we have a lot of mitochondria, because the generation and transmission of impulse needs a lot of energy. Also, we have lysosomes here, because lysosomes provide intracellular regeneration for neurons. Every neuron lives as long as we live. So the age of our neurons is equal to our age. They cannot divide, so we have no stem cells for them, but they should regenerate their organelles. And when organelles become too old and not very effective, they should be destroyed with the formation of new organelles. And the old defective organelles are destroyed with the help of lysosomes. So in neurons we have usually also a lot of lysosomes to destroy old defective organelles. And also we have cytoskeleton, which includes neurotubules or microtubules of neurons, neurofibrils, which neurofilaments, which are intermediate filaments, and microfilaments. They help to maintain the shape of neuron with all its processes, and also microtubules or neurotubules help to perform the transport of organelles and chemicals along the long processes of neurons. As you can see here, the protein synthesizing apparatus with rough endoplasmic reticulum is present in the perikaryon, in the cytoplasm of the cell body, and also in dendrites, but not in the axon. The initial segment of axon is called axon hillock. This is the area where the response of a neuron is generated. So a neuron receives information from other neurons along its dendrites, then this information is processed, it is summarized, and some other functional processes happen, and then in axon hillock the membrane decides whether there will be the impulse or not. And if there will be the impulse, then it will go along the membrane of the axon. And axons are usually covered with myelin sheets. We have some specific stainings for nervous tissue. One of them is the so-called Nissl stain. Nissl is the name of the scientist who invented this type of staining. This is the stain which stains acidic structures. So it is the analog of hematoxylin, and it stains DNA in the nucleus, and also nucleolus is stained. And what is more important, in the cell body, the Nissl bodies are stained. Nissl bodies are the complex of well-developed rough endoplasmic reticulum, polyribosomes, and free ribosomes. In this diagram, you can see these Nissl bodies, these dark patches, and one of them is shown here how it looks like in transmissional electron microscopy. And as you can see here, one of these structures, one Nisl body consists of several cisternae of rough endoplasmic reticulum and some polysomes and free ribosomes. So this is the element of protein synthesizing apparatus. And as you can see here, all the cytoplasm is filled with this protein synthesizing apparatus, which is called Nisl bodies. And Nisl bodies are found in initial segments of dendrites, so dendrites are visible in this staining, but they are absent in axon and its axon hillock. So axons cannot be visible in this staining. So the processes which we see are dendrites. Axons are not visible. In this picture, you see the part of the section of the spinal cord. So here you can see some neurons. In some of them you can see nuclei, in some of them we do not see nucleus. This is because of the sectioning. And you can see here a lot of cell nuclei. All of these cell nuclei are the cell nuclei of glial cells. Everything which is between the neurons and glial cells is called neuropil. It consists of processes of neurons and glial cells. So in nervous tissue we have almost no extracellular matrix. So all the space is filled with processes of neurons and glial cells. Another example of this type of staining, so here we can see the neurons stained with Nisl staining. Nisl bodies are found in pericardion and in dendrites. Axons are not seen. These are cell nuclei of glial cells. And here, this is the electron microscopy picture of the part of cytoplasm of the neuron. Here is its nucleus with nucleolus and a part of cytoplasm inside which we see Nisl bodies. 1, 2nd, 3rd, etc. One Nisl body consists of several rough endoplasmic reticulum cisternae and a lot of polyribosomes. We will see this type of staining in the preparation of the cross-section of spinal cord stained with Nisl stain. So this is the spinal cord. The area which includes the cell bodies of neurons is called gray matter. It contains cell bodies of neurons. It is butterfly-shaped area. And this area doesn't contain any cell bodies of neurons, and this is called white matter. Another example of this preparation: gray matter with cell bodies of neurons, white matter. The same as here. Second type of staining for nervous tissue is the silver stain. Silver stain stains neurofibrils inside the cytoplasm of the cell, both in pericardion and in dendrites and axons also. Neurofibrils are the combination of neurotubules, which are microtubules, and neurofilaments, which are intermediate filaments. So microtubules and intermediate filaments together form neurofibrils. Neurotubules and neurofilaments cannot be seen in light microscopy picture. They can be seen only in electron microscopy pictures. So these are thicker neurotubules and thinner neurofilaments. But when they are stained with silver stain, they form very thick neurofibrils, which are so thick so they are seen in light microscopy picture. So here you can see this type of staining, and we see these neurofibrils both in cell bodies and in processes. As I said previously, microtubules are not only for maintaining of the shape of a neuron, but also they perform the transport, especially axonal transport system. So microtubules serve as the railway for transporting of vesicles containing neurotransmitters, for example, some vesicles, and organelles. How this happens? We have the carrier proteins such as dynein and kinesin. These proteins have two ends. One of this end can walk along the microtubule in one direction, and second end of this molecule can be attached to some membranous organelle, for example mitochondrium seen in this picture. So the dynein protein attaches with one end to microtubule and with the second end to mitochondrium, and then it walks this mitochondrium from the periphery, from the tip of the axon towards the cell body. And kinesin does the same, but on the opposite direction, in the opposite direction, from the cell body towards the periphery. So we have retrograde. Retro means backwards. It means back from the periphery to the cell body. And anterograde, which means from the cell body towards the end of the axon. Anterograde transport is subdivided into fast and slow. Fast is about 100-500 millimeters per day, and it allows transportation of endoplasmic reticulum, cisternae, mitochondrium, and neurotransmitter containing vesicles from the cell body, where they are generated, to the tip of the axon, where the synapse is located. Synapse is the area for transmission of nerve impulse from one neuron to other neuron. So we need these organelles here. We need here mitochondria, endoplasmic reticulum, and neurotransmitter containing vesicles, but they are generated inside the cell body. So they should be transported to their place of functioning via the anterograde transport. And we have slow transport. It is the transportation of cytoskeletal elements and enzymes, which are needed for maintaining the shape of the axon and for its growth. Retrograde transport performs backwards from the periphery to the soma, and it serves for removal of too old and damaged elements from the axon terminals towards the cell body, where they will be degraded with the help of lysosomes. So it ensures turnover of vesicles and mitochondria. So mitochondria which become too old should be returned to the cell body, where they will be degraded with lysosomes, and new formed mitochondria will be transported backwards via anterograde transport. Here you can see the cross-section of spinal cord stained with silver impregnation. As in Nissl staining, we see here gray matter with cell bodies of neurons, and white matter where we have no cell bodies of neurons. The same is here. So in gray matter we have cell bodies of multipolar neurons. All neurons here are multipolar. Also axons and dendrites of neurons, and also glial cells, which include astra, oliga, microglia, and also blood vessels. In white matter we have cross-sections of myelinated nerve fibers, glial cells, including astra, oliga, and microglia, and blood vessels. Here you see the silver impregnation of spinal cord at high magnification. We see here cell bodies of neurons, inside which we see neurofibrils which are stained with silver. This is neuropil, which contains processes of neurons and glia, and cell nuclei of glial cells are almost not seen. The same is here. Cell body, processes of a neuron, its nucleus, cutted sections of processes of other neurons in neuropil, and not stained nuclei of glial cells. So two types of specific stainings for nervous tissue. Silver impregnation reveals neurofibrils, and methylene blue or Nissl staining reveals Nissl bodies. Now, in nervous system, we have central and peripheral nervous system. Central nervous system includes brain and spinal cord, and peripheral or all the rest, which is nerves and ganglia. Ganglia are the groups of neurons outside of the CNS. So we have neurons not only in brain and spinal cord, but also outside brain and spinal cord, in some ganglia, and also inside the organs. Now we discuss glial cell types. Glial are accessory cells. They help neurons to perform their function. And glial cells are different in CNS and PNS, which means central and peripheral nervous system. In central nervous system, we have microglia, the glial macrophages, and macroglia. Macroglia includes astrocytes, ependymal cells, and oligodendrocytes. They have different sources of development. Glial macrophages develop from red bone marrow, and in embryonic development they develop from mesenchyme. The macroglia cells, astrocyte, ependymal cells, and oligodendrocytes develop from neural tube. Now, in peripheral nervous system, we have two types of glial cells: Schwann cells or neurolemocytes, and mental gliocytes. Both develop from neural crest. In this picture, you can see the diagrams of all the glial cell types. In the last picture, you see two glial cells in PNS: mental gliocytes, which surround cell body of a neuron, and Schwann cells here. In all the other pictures, you can see glial cells in the central nervous system. They include two types of astrocytes: protoplasmic and fibrous astrocytes. Protoplasmic are located in gray matter, and fibrous in white matter. Oligodendrocytes, also the glial cells in the brain and spinal cord. Three types of ependymal cells: ependymal cells proper, tenicytes, and choroid ependymocytes, and microglial cell. The same is in this picture, and now we will discuss them in details. So, astrocytes. We have two types of them: protoplasmic in gray matter and fibrous in white matter. Here are their functions. They provide the functions of architectonic. It means they make the three-dimensional scaffold for all other cells in CNS. They have star-like shape with their processes, which maintain the shape of nervous tissue in CNS. Next, barrier function. Some of the processes of astrocyte form the end feet, large end of the process, which form here in this picture perivascular membrane. Peri means surrounding, vascular means blood capillary. So these end feet of astrocyte cover all the blood capillaries. Here is the blood capillaries, and form the blood-brain barrier, the barrier between the blood in blood capillaries and nervous tissue surrounding it. And also some of the processes of astrocytes, which are located closely to the periphery of the nervous tissue in brain and spinal cord. These processes also form the end feet, which cover the nervous tissue from the outside and form the glial limiting membrane. It covers the nervous tissue from outside and separates nervous tissue here from the loose fibrous connective tissue of the pia mater, which covers the brain from outside. Next, trophic function. Astrocytes, through their end feet, receive the substances from blood capillaries, and then they transmit these substances to neurons. So they feed neurons. Also, astrocytes are the part of synapse. They take some excessive ions and neurotransmitters from extracellular matrix and um, give these ions and neurotransmitters back to neurons. And also, they can form a glial scar in the case of brain injury and death of neuron. If a neuron dies, it cannot be replaced by other neurons, and the area previously filled with neuron after its death will be filled with astrocytes, which can divide, they can proliferate, and they can migrate to the areas of injury. And also, they surround the damaged area and form the dense scar tissue. Here you can see the structure of blood-brain barrier. It's the barrier between the blood in the capillary and the neurons in nervous tissue. So its barrier includes the endothelial cell of the blood capillary, its basement membrane, and the end feet of astrocytes. Here are end feet of astrocytes and astrocytes themselves, which are connected to the neurons. So everything which passes from blood to neurons should pass through the cytoplasm of astrocytes, who decide whether or not this particular substance is toxic to neurons. Next, oligodendrocytes. Oligo means few, dendro means processes. So unlike other glial cells, astrocytes, oligodendrocytes have fewer amount of processes, so this is why they are called so. They are located both in gray and white matter in brain and spinal cord. They participate in formation of myelinated and unmyelinated nerve fibers. Their processes wrap around the axons of neurons and form the covering, which is called myelin sheath. This myelin sheath acts as the insulator, which allows the faster transduction of nerve impulse along the axon of the neuron. So oligodendrocytes modulate conduction of nerve impulses along the axons. Also, they participate in repair of neuronal damage by phagocytosis of distal portion of the damaged axons and myelin. And also they perform insulation, trophic function, architectonic, and protection function as other types of neuroglia. Next, we have three subtypes of ependymal glial cells: ependymocytes proper, tenicytes, and choroid ependymocytes. Ependymocytes proper form the single cell layer which surrounds central canal in spinal cord and also lines the brain ventricles, which are connected to the central canal, the cavities inside the brain filled with the cerebrospinal fluid. So these cells have the cilia in their apical surface, and these cilia help to move the cerebrospinal fluid along the ventricles and central canal in spinal cord. Next, tenicytes are the subtypes of ependymocytes. They have a lot of microvilli and some single cilia. They have one long process which wraps around the blood capillary. So this type of cells, which are located in the third and fourth brain ventricles, they help to capture substances from liquor or the cerebrospinal fluid, um, and transfer them to blood capillary. So they perform communication between the cerebrospinal fluid and the blood. And finally, choroid ependymocytes. They are cuboidal cells located upon the basement membrane with a number of cell junctions, including tight junctions, so they resemble epithelial cells. They are found in the so-called choroid plexuses in the brain. These are the areas where the cerebrospinal fluid is made from the blood plasma via the filtration through these cells. So blood plasma is filtrated through the uh, blood liquor barrier and becomes the liquor or cerebrospinal fluid. We will see the ependymal cells, which line the central canal in the histological preparation of cross-section of spinal cord in Nissl and also in Silverstein. So this is the central canal. It is filled with cerebrospinal fluid, and it is lined with one layer of ependymal cells, which have cilia upon their apical surface and one long basal process. The choroid ependymocytes are seen in this picture of blood liquor barrier, which is the barrier between the blood in blood capillaries and liquor or cerebrospinal fluid. This barrier is where the choroid plexus is located, so this is the cross-section of choroid plexus. It contains loops of blood capillaries filled, surrounded with loose connective tissue and covered with one layer of choroidal ependymal cells. So here you can see these cuboidal choroid ependymal cells through which blood plasma is filtrated to become the cerebrospinal fluid. Microglia uh, is actually the glial macrophage. It's the part of the macrophage system of the body. In resting state, it looks like this, but if it is activated, it becomes the macrophage-like. It has the pseudopodia, it can move, and it performs phagocytosis of damaged neurons and other damaged or dead cells in CNS. Here you can see the picture showing cells in central nervous system. In the left part, uh, here is the gray matter, and in the right part, white matter. In gray matter, we have cell bodies of neurons, blue in this picture. They are surrounded with astrocytes. Here is the astrocyte, uh, which forms end foot, which covers the blood capillary and forms blood brain barrier. Here we have microglial cell, and these are oligodendrocytes, which have processes which form myelinated sheets, which surround the axons of neurons to form myelinated nerve fibers. Also, we see here the part of the brain ventricle, or maybe central canal in spinal cord, which is surrounded or lined with the ependymal cells. Another example of the same picture. So here we have here the uh, part of the brain. It is covered with the pia mater, which includes loose fibrous connective tissue with blood vessels, and the nervous tissue is separated from the pia mater with the help of the processes of astrocytes, which form the limiting membrane here. And also astrocytes form the perivascular membrane, which covers the blood vessels. Oligodendrocytes, here we have oligodendrocyte, it forms the myelinated uh, sheet around the axon of the neuron. Microglia is a small cell here and here also, and ependymal cells with cilia line the ventricle of the brain. Here you can see how the glial cells, astroglial cells, cover the area of synapses, the contacts between the two neurons through which the impulse goes from the first to the second neuron. So all this area is surrounded with the processes of astrocytes. So in our histological preparation of spinal cord in cross-section, we will see ependymal cells around the central canal filled with the cerebrospinal fluid. In gray matter, we will see multipolar neurons and glial cells, their nuclei, which include astrocytes, protoplasmic in gray matter, oligodendrocytes, and microglia. We cannot differentiate between them, but we should know which types of glial cells can be found in gray matter. Now, in peripheral nervous system, we have two types of glial cells: mental glial cells, which surround cell bodies. In ganglia, this is the cell body of a neuron, pseudo-unipolar. Second type of glial cells in PNS is called the Schwann cell, after the name of the scientist, or other name is neurolemmocyte or just lemmocyte. These are the cells which, like oligodendrocytes in CNS, form the myelin sheaths around the axons. We will see these types of cells in preparation of dorsal root ganglion. It is the sensory ganglion. Ganglion is the group of neurons outside the central nervous system. One of such ganglia are sensory dorsal root ganglia. So here we see pseudo-unipolar neurons. Their axons go to spinal cord, and their dendrites receive information from the periphery, from the receptors. The cell bodies of neurons are surrounded with mental glial cells, and the processes of pseudo-unipolar neurons form nerve fibers with the help of Schwann cells. So we will see here one type of neuron, pseudo-unipolar neuron, and two types of glial cells, mental cells around the cell bodies and Schwann cells around the processes of neurons. Here is this preparation at higher magnification. Cell bodies of neurons are seen here, and all the rest area is filled with nerve processes. High magnification, cell bodies of neurons rounded here. All this organ is covered with a capsule of dense fibrous irregular connective tissue, and these areas are filled with nerve fibers, with Schwann cells, and some amount of loose connective tissue between the nerve fibers. At high magnification, what do we see here? Cell bodies of pseudo-unipolar neurons, a cell nucleus of this neuron, nucleolus in the middle. Every pseudo-unipolar neuron is surrounded with mental glial cells, which have round cell nuclei. So the cell nuclei which are very close, located very tightly to the cell body of a pseudo-unipolar neuron, are the mental glial cell nuclei. If we go further from the neuron, we will see other cell nuclei. Some of them are flattened and elongated. They are cell nuclei of loose fibrous connective tissue, which surrounds neurons and nerve fibers. So we should differentiate here between mental glial cells, which are the closest to the cell bodies and round in shape, and cell bodies of loose fibrous, cell nuclei of loose fibrous connective tissue cells, which are further from neurons and have more flattened shape of cell nuclei. So here we will have nerve fibers which grow from the cell bodies of neurons, and they are accompanied with the Schwann cells. All this organ is covered with a capsule of dense fibrous irregular connective tissue, red in color because of bundles of collagen fibers. Here are the cell nuclei of fibrocytes, and here some amount of loose fibrous connective tissue and maybe adipose tissue here. So at the highest magnification, some cell bodies of pseudo-unipolar neurons surrounded with mantle glial cells. Further from neurons and with more flattened nuclei, cell nuclei of loose fibrous connective tissue cells, and also some blood capillaries inside it. And now we go to nerve fibers. Nerve fibers are nerve cell process which can be axon or dendrite surrounded with glial sheath. So we have two types of nerve fibers, unmyelinated and myelinated. Both types contains two components, a process of a neuron, which is called axon cylinder number 1 here. It can be either axon or dendrite. And second component, the glial cell, which surrounds the process of a neuron in both cases, in the case of myelinated and unmyelinated too. But in the case of myelinated nerve fiber, the glial cell wraps many times around the central cylinder to form the so-called myelinated sheath, number 4 here. And myelinated sheath acts as the physical insulator for the electrical impulse and allows myelinated fibers to have more high speed of the nerve fiber transduction. So the speed of nerve of electrical impulse transduction through the unmyelinated fiber is less than 2 meters per second, but in myelinated ones more than 100 meters per second. Unmyelinated nerve fibers are present only in autonomic nervous system, only in peripheral nervous system, and they form the postganglionic nerve fibers in autonomic nerve system. These fibers are the axons of the efferent neurons which innervate some tissues in our organs, such as smooth muscle tissue in stomach and intestines, or glandular cells, the glands which produce some substances. We do not need very high speed of innervation of such tissues. So we do not need these fibers to be myelinated. But myelinated fibers, they are found in CNS and PNS. They are forming all the nerve fibers in somatic reflex arcs which innervates skeletal muscles, and they form preganglionic nerve fibers of autonomic nervous system. So our somatic reflex arcs which innervate skeletal muscles should work very fast, because we need to contract our muscles voluntarily very fast, so we need here very fast myelinated nerve fibers. So here we see here two pictures. The upper shows the myelinated nerve fiber and lower unmyelinated. In the case of myelinated nerve fiber, we have the axon of the neuron, which is covered with a series of Schwann cells with some areas between them called nodes of Ranvier. So this is the area of the axon not covered with the myelin sheath. Myelin sheath acts as the insulator, so no opening of ion channels can be performed in the area of the axon covered with the myelin sheet. And so the wave of depolarization spreads saltatorily. So it means the impulse jumps from one node of Ranvier to other one, to the next one. And this happens very fast. But in the case of myelinated axon, the action potential moves very slow along the membrane, like this. So unmyelinated nerve fiber, here you see how it is formed. So axon or dendrite of the neuron axis cylinder is surrounded with a glial cell, which wraps around this axial cylinder, and here you see where the membrane of the cell meets other portion of the membrane, forming the so-called mesaxon here. And thus we have the axis cylinder covered with the glial cell from all the sides. But it is not inside the glial cell itself. It is not inside the cell. It is just covered with the cell from the outside. This is how such unmyelinated nerve fiber looks like. One glial cell can cover more than one axial cylinder. Usually we have several axial cylinders, rounded structure in this picture. So here we have 1, 2, 3, 4, 5, 6, 7 axial cylinders in the structure of one unmyelinated nerve fiber. This is the cell nucleus of the glial cell, the Schwann cell. Here is its cytoplasm. Here is the mesaxon, this area where the membrane meets itself. And so each axial cylinder is covered with the glial cell. Now, myelinated nerve fiber, it starts to be formed as previously we have seen in myelinated one. So axial cylinder is surrounded with the glial cell. Glial cell, the membrane of glial cell meets itself here and forms mesaxon, but then it continues to wrap and wrap and wrap around the central cylinder. And this is what happens. We have elongation of mesaxon, and the layers of mesaxon, layers of membrane of the Schwann cell finally meet together to form many layered structure which consists of membrane, cell membrane of the Schwann cell, and which is called myelin sheet. And the cytoplasm of the Schwann cell goes to the periphery. It is called here neurolemma. And if we look here at the longitudinal section, so in the center we have the axial cylinder. This is the myelin. It consists of several layers of the membrane of the Schwann cell. We have here also the so-called Schmidt-Lantermann clefts, which go oblique. This is the node of Ranvier, the area where the one Schwann cell ends, and next will be the second Schwann cell. So node of Ranvier is the area between two myelin sheets made up by two Schwann cells. Here you can see that the processes of one and second cells are intersecting and covering this area from outside. And here you can see that the cell nucleus of the glial cell, the Schwann cell, is located at the periphery of the nerve fiber. We have myelinated nerve fibers both in peripheral and central nervous system, and here are some differences. First, in PNS, the myelin sheet is made up with the help of a Schwann cell. in CNS with the help of oligodendrocyte. Second, in PNS, the Schwann cell um, wraps around the axon, including all its cytoplasm and including its nucleus. So we will see the cytoplasm and the nucleus of the Schwann cell included in the nerve fiber at its periphery. But in CNS, the cell body and cell nucleus of oligodendrocyte uh, doesn't participate in the myelinating sheet of the axon. It stays outside of the myelin sheet. Only process of the oligodendrocyte goes towards axon, wraps around it, and forms the myelin. Next, in PNS, we have these oblique Schwann-Lantermann uh, clefts and no such clefts in CNS. And also in the peripheral nervous system, the nerve fibers are covered with basement membrane from the periphery and no basement membrane in CNS. We use specific osmic acid staining to see the myelin in nerve fibers. Osmium acid or osmic acid stains lipids because myelin, as you see here, is actually made up with many layers of cell membrane, and the cell membrane consists of phospholipids. These phospholipids are stained with osmic acid in black color, and thus we see the myelin which looks black. Black structure surrounding the non-stained axial cylinder in the center. So here we see the drawing of the preparation stained with osmic acid and the carmine staining to see cell nuclei. In both cases, we have the cell nuclei of Schwann cells included in the myelinated and non-myelinated nerve fibers in peripheral nervous system. Uh, in this diagram, we see it in cross section. So, in myelinated nerve fiber, we have the axon or axial cylinder in the center. Here it is non-stained. It is surrounded with myelin sheet. It includes many layers of membrane of the Schwann cell. Myelin is stained in black color. The cytoplasm and the cell nucleus of the Schwann cell, or lemma site, is located at the periphery, number 5 in this picture, at the periphery, outside of the myelin itself. And also we see here that this myelinated fiber is covered with a basement membrane from the periphery, and also we have here the Schmidt-Lantermann clefts, so this is the myelinated nerve fiber of the peripheral nervous system. In unmyelinated nerve fibers, we do not see myelin, so they look very pale. Only cell nuclei are seen because they are cross stained with carmine in this drawing. So here we see the cell nucleus in this diagram, cell nucleus of the Schwann cell, its cytoplasm, and the axial cylinder, maybe axons, maybe dendrites, which embed into the glial cell. They kind of hang on the mesaxon, made up with the cell membrane of the Schwann cell. So here we see the cell nuclei of Schwann cells and cytoplasm of Schwann cell, and the axons are not stained in osmic acid staining. Here you see the transmissional electron microscopy pictures of cross-sections of unmyelinated and myelinated nerve fibers. In the left picture, we see only unmyelinated nerve fibers. In this one, in the center, we see cell nucleus. This is the cell nucleus of the Schwann cell. It is surrounded with cytoplasm of this Schwann cell, and these These lighter and round structures are the cross sections of the axial cylinders of the processes of neurons. So these can be axons of dendrites of neurons. They are surrounded with the cytoplasm of the Schwann cell. In these sections, we do not see cell nuclei of glial cells, but all other components are present. We see rounded and more light sections of axial cylinders of neurons surrounded with grayish cytoplasm of the Schwann cell. And from the outside, we see the basement membrane. And the same in these sections. In the picture on the right, we have one large myelinated nerve fiber. In its structure, we see the cell nucleus of the Schwann cell, the cytoplasm of the Schwann cell here, the myelin sheath itself, very black structure. It is made with the several layers of the membrane of this Schwann cell, and we see the axial cylinder in the center. This is the mesaxon. Inside the axial cylinders, those dots are microtubules, and this is mitochondrium. The same as here, small dots are microtubules, these are mitochondria. In the picture on the right, we have also some sections of unmyelinated nerve fibers, such as this one. It also includes several axial cylinders surrounded with the cytoplasm of the Schwann cell. Mesaxons are seen here, and here also. In the section below, also we see several axial cylinders surrounded with cytoplasm of glial cell, and mesaxons are seen here. In this electron microscopy picture, we see the sections of myelinated, here with black myelin sheets, and unmyelinated nerve fibers at low magnification. Between individual sections of nerve fibers, we see lighter areas. These are the areas of loose fibrous connective tissue filled with the fibers of loose connective tissue, and this is one cell of loose fibrous connective tissue, maybe a fibroblast. In this picture, we see the cross-section of the peripheral nerve stained with hematoxylin and eosin staining. So, here we see the bundle of nerve fibers. It consists of several myelinated and unmyelinated cross-sected nerve fibers. In hematoxylin and eosin staining, myelin itself is not stained. It stays as unstained light area. But here, the cell nuclei and the axial cylinders are stained. So, for example, here we see the dot in the center. This is the axial cylinder. It is surrounded with non-stained area myelin, and at the periphery we have the cytoplasm of the Schwann cell. The same at high magnification. This is the example of one myelinated nerve fiber. We have the axial cylinder here. It is stained. It is surrounded with non-stained area of the myelin itself. And at the periphery, we have the cytoplasm and the nucleus of the Schwann cell. The same as here. Axial cylinder, myelin, cytoplasm of the Schwann cell. Between nerve fibers, we see loose fibrous connective tissue, and here should be the cell nucleus of some cells of loose fibrous connective tissue. This is the preparation of the peripheral nerve with myelinated and unmyelinated nerve fibers stained with osmic acid and carmine for cell nuclei. We see here both thick and dark myelinated nerve fibers and thin and light unmyelinated nerve fibers. In unmyelinated nerve fibers, we see pinkish cell nuclei of Schwann cells. In myelinated nerve fibers, we see a lighter area in the center, this is the location of axial cylinder, and darker areas surrounding it. These are the areas of the myelin sheath. Also, we should see some cell nuclei of Schwann cells, but not in this picture, maybe this one. The Schwann cell nucleus in the structure of the thick myelinated nerve fiber. Also, we see here the node of Ranvier, the area where one Schwann cell ends and second Schwann cell begins, the area between two Schwann cells. The same here at high magnification, thick myelinated nerve fiber, node of Ranvier, in the middle of the nerve fiber, axial cylinder, at the periphery, more dark myelin itself. Unmyelinated nerve fibers, very pale, with the cell nuclei of Schwann cells, and thick myelinated nerve fibers. Unmyelinated, myelinated nerve fibers. In this picture, we see here the cell nucleus of the Schwann cell in the structure of myelinated nerve fiber. This is the cross-section of the peripheral nerve, also stained with osmic acid, and we see here myelinated nerve fibers with very dark myelin sheath and not stained axial cylinder in the center, and between them unmyelinated nerve fibers and loose connective tissue, the structures which are more pale in color. So in high magnification, we see here myelinated nerve fiber. It is the myelin itself which is stained with osmic acid, and axial cylinder which is in the center is not stained. This is the area of unmyelinated nerve fibers and loose fibrous connective tissue. It looks more pale. Now, in peripheral nervous system, axons can be regenerated. Here you can see the cell body of the motor neuron. Its axon goes towards the muscle fiber and innervates it through the motor plate. Axon is covered with the Schwann cells, which make the myelin sheath around it. In the cytoplasm of a motor neuron, we have a lot of Nissl bodies, which are groups of rough endoplasmic reticulum and polysomes. If we have the injury, if our nerve is cut, then the distal portion of the axon will be degraded, it will die, and its remainings of the Schwann cells will be destroyed and eaten with macrophages. Macrophages come here from the surrounding loose connective tissue. So this part will be destroyed and eaten with macrophages. The muscle fiber, which doesn't receive impulse from neuron anymore, becomes hypertrophied, it becomes smaller and non-active. In the cell body of a neuron, the Nissl bodies degrade. But the proximal part of the axon still remains alive, and the Schwann cells here also are alive. Schwann cells, glial cells, can proliferate, so they proliferate, they divide, and they go along the covering of the nerve towards the muscle fiber, and they form the bridge from the proximal part of the nerve towards the muscle fiber. And then the end portion of the axon start to branch and grow along this bridge made up with glial cells. And if everything is good, then the axon reaches in the end its muscle fiber, and forms motor plate once again, and now this muscle fiber receives the impulse and grows again and becomes normal again. But if the ends of the proximal part of nerve and distal part are separated from each other, then the bridge cannot be formed and the axon cannot reach the bridge through which it can reach the muscle fiber. So it just stays here and forms the amputation neuroma. And uh, now the muscle fiber will not receive the impulse from this neuron, never. Now, reflex arc is the chain of neurons connected by synapses and providing nerve impulse from the receptor of a sensory neuron through the interneuron towards the effector neuron and through its axon to the effector endings. So, general principle of reflex arc organization. Here in this picture, you see that we have three neurons. In general, in reflex arc, first one is sensory or afferent neuron, second one is interneuron or associative neuron or relay neuron, and third one is effector or motor or efferent neuron. So, also we have different types of nerve endings. The ends of dendrites of sensory neurons are called receptors, and they are subdivided into free and non-free, which are subdivided into encapsulated and non-encapsulated. So they form afferent nerve endings which receive stimuli. Then impulse goes from receptors, ends of dendrites, along the dendrites of sensory neurons towards its cell body, and then from the cell body along the axon to the second neuron. And then we will have the synapse, the connection between the end of axon of the first neuron and dendrite of the second neuron. Then impulse goes towards the cell body of the second neuron and along its axon to the third neuron. Also, we will have here synapse between the axon of the second neuron and the dendrite of the third one. And then the impulse goes along the axon from the cell body to the end of the axon of the effector neuron, and the axon of the effector neuron can end with motor plate in the case of the innervating of skeletal muscles, or secretory nerve ending in the case of innervating smooth muscles or glands. So these are efferent nerve endings, endings of axons of efferent neurons. So here we have three types of reflex arcs: somatic, which innervates muscle fibers, this is muscle fiber, and B and C stands for autonomic reflex arcs, which innervate our organs. They are subdivided into sympathetic, number B, and sympathetic, number C. In these pictures, they innervate smooth muscle cells. So in all three pictures, we have three neurons. Neuron number 1 is located inside dorsal root ganglion. It is the sensory neuron. Neuron number 2 is located inside spinal cord. It's the relay associative neuron, and here also it is located. And the third one in the somatic reflex arc, it should be here. In this picture we have only two neurons. The associative is missed here. So here we have efferent neuron inside the brain in the somatic reflex arc, but in both sympathetic and parasympathetic, the third neuron is located outside of the spinal cord at the periphery in the peripheral autonomic ganglion. Ganglion is the group of neurons outside of the CNS. So in the sympathetic one we have sympathetic ganglion, and in parasympathetic we have parasympathetic intramural ganglion, which is located inside the um, wall of some organ. The dendrite of the pseudounipolar neuron number 1, the dendrite ends with receptor. The axon ends with the synapse. Synapse is the connection between the axon of one neuron and dendrite of second one. And the end of the axon ends with efferent ending. In this case, it is called motor end plate because it innervates skeletal muscle fiber. In the case of both sympathetic and parasympathetic, the efferent endings are of secretory type. They have the thickening areas of the axons filled with vesicles with neurotransmitters, which go outside when the impulse wave reaches this area and act upon the receptors on the surface of smooth muscle cells. So nerve endings are sensory receptors, interneurons, synapses, and efferent endings. Synapses are the connections between two neurons. They usually include the end of axon of one neuron and the dendrite of the second neuron. We have two types of synapses, electrical and chemical ones. Electrical resemble gap junctions, nexuses, so they look like nexuses. They have the channels through which the ions can go in both directions, from the first neuron to second one and from the second one to the first one. Electrical synapses are very fast, but they are non-specific. It means that they can act in both directions. They are present only during embryogenesis. In the adult organism, we have chemical synapses. They are very specific. They transmit impulse only from neuron 1 to neuron 2, and the impulse is transducted with the help of neurotransmitters. So, what do we see here? This is the ending of the axon of the first neuron. This is the area of the axon covered with myelin sheet. So in the ending, we have no myelin covering the axon. Inside axon, we have microtubules along which vesicles with neurotransmitters reach this area. And in this presynaptic area, enlargement of the end of the axon, we have a lot of synaptic vesicles filled with neurotransmitters. This is the area between the axon of the first neuron and the dendrite of the second neuron. This area is called synaptic cleft. And the third component of the synapse is the postsynaptic part. This is the membrane of the postsynaptic neuron, the second neuron, inside which we have the receptors to this particular neurotransmitter. So what happens when the wave of depolarization of the membrane reaches this area? It reaches the area with calcium channels, which are voltage sensitive. It means that they can sense the change of the charge of membrane, and when it happens they open and calcium goes inside the cell, inside the presynaptic terminal. And calcium causes the exocytosis of the synaptic vesicles, and neurotransmitter goes out from the axon into the synaptic cleft. Then this neurotransmitter diffuses through the synaptic cleft to reach the receptors, and when neurotransmitter binds to receptors, it causes the opening of the ion channels in the postsynaptic membrane, and this will cause the generate of action potential on this membrane, and then this action potential will run along the membrane of the second neuron. So these steps are shown in this picture. So first, the polarization of presynaptic membrane causes opening of calcium channels. Calcium goes into the cell. It causes exocytosis of synaptic vesicles with neurotransmitters. Neurotransmitters react with receptors and provide depolarization of the postsynaptic membrane. Here is other picture of the synapse: presynaptic part, synaptic cleft, and postsynaptic membrane. In presynaptic part we have microtubules, smooth endoplasmic reticulum, mitochondria, and vesicles with neurotransmitters. These are proteins which ensure exocytosis when calcium comes into the cell. In the synaptic cleft we have the proteins which attach the membrane of the first neuron to the membrane of the second neuron. And in the postsynaptic part we have the elements of cytoskeleton, which help the transduction of impulse and help to maintain the shape of the postsynaptic membrane. According to the location of the synapses, we have the exodendritic synapse if it is formed with the axon of first neuron and the dendrite of the second one. We can also have axosomatic synapse if it is formed with the axon of one neuron and the soma or cell body of the second one. And finally we can have the axonal synapse if it is formed with the axon of one neuron and axon of the second one. This type of synapse is usually inhibitor. Efferent endings are motor end plates in the somatic nervous system and secretory endings in autonomic nervous system. Somatic nervous system innervates skeletal muscles. Autonomic nervous system innervates smooth muscles and glands in our body. So it should not be very fast, unlike somatic one. In somatic nervous system, efferent ending is the motor plate, the area of the direct contact between the end of the axon and the muscle fiber. The neurotransmitter here is the acetylcholine. This ending triggers skeletal muscle contraction. It means no nerve impulse, no contraction. We need nerve impulse from neuron for contraction. Without it will be no contraction. But in autonomic nervous system, in secretory endings do not have specific contacts with the target cells, and they do not have a triggering function. It means that smooth muscles can contract without different innervating from the nervous system. But the nervous system thus just regulates the activity of the smooth muscles, secretory cells, and also cardiac muscle cells. So secretory ending are for just regulating or modulating the contraction mode. It means the strength of contraction and the speed of contraction. In the secretory endings we have neurotransmitters such as acetylcholine and norepinephrine or noradrenaline. Here you can see the structure of motor end plate. This is the direct synapse between the neuron and muscle fiber. Here you see the end of an axon. It lacks the myelin sheet in this area. So here we still have the myelin, here we do not have myelin sheet. In the presynaptic part, we have a lot of synaptic vesicles with neurotransmitter acetylcholine, and this is the synaptic cleft. This is the area between the membrane of the axon and the membrane of the muscle fiber. I mean sarcolemma. Sarcolemma includes the cell membrane and the basement membrane, which is grayish in this picture. So we have the larger one, primary synaptic cleft, from which smaller secondary synaptic cleft grow. When impulse reaches this area, influx of calcium ions trigger exocytosis of vesicles with acetylcholine. Acetylcholine diffuses through the synaptic cleft to reach the receptors located upon the membrane of the muscle fiber, and this causes opening of sodium channels and generating and spreading of the nerve impulse along the membrane and then through the T-tubules to the sarcoplasmic reticulum, where it causes releases of calcium, which reaches the actin myofilaments and which triggers the contraction of myofibril. In this picture, you see the axon terminal marked with label T with a lot of small rounded vesicles with acetylcholine. This is the synaptic cleft. This one is primary, and these are secondary. Inside them, the grayish thing is the basement membrane of the muscle fiber. This black line is the cell membrane of the muscle fiber, and here M stands for muscle fiber cytoplasm, sarcoplasma. The same as here in this picture, and this also the same. Receptors are the dendrite endings of sensory neurons. They are free or non-free according to the presence of glial cells. If there are no glial cells surrounding the end of dendrite of the sensory neuron, this receptor will be called free. If the end of dendrite is surrounded with glial cell, it is called non-free. And non-free are subdivided into encapsulated or unencapsulated, based upon the presence of outer surrounding connective tissue. So if there is the outer capsule, there will be the name encapsulated. If no other surrounding, we will have unencapsulated receptors. Here you see these receptors. In the first picture, we see the free nerve ending, which receives the sensation of the pain and temperature. This is the end of the dendrite. It has a lot of branches. It is located in the skin. So this is the epidermis of the skin. This is the connective tissue of the dermis. This is the basal layer of epidermis. This is the stratum spinosum. And you see how the dendrite reaches the basal layer of epidermis, and the thin branches of dendrite go between the cells in the thickness of the epidermis. And these free endings of the dendrite can easily receive the pain and temperature information. Next, we have the Merkel disc, the touch receptor. Merkel cell is one of the accessory cells in the epidermis, in the stratified squamous keratinized epithelium. Such cell is also located in the basal layer, and here you see the end of the dendrite, which reaches this cell to receive the information from this cell. It is called Merkel disc, touch receptor. Also in skin we have Meissner corpuscle. This is the non-free encapsulated nerve receptor. It is non-free because it has the covering of glial cells. These are glial cells. They cover the end of dendrites. And also we have outer capsule made up with the specific connective tissue cells. These corpuscles are located in the papillae of the dermis, just below the epidermis, and the outer capsule is connected with the collagen fibrils with the basement membrane of the epidermis. And very fine touches to the skin causes the excitation of the dendrite of this Meissner's corpuscle, so it serves for touch reception. Next we have Pacinian corpuscle. It receives the pressure and vibration. It also can be found in skin, but in lower areas where we have the border between the dermis and hypodermis, where dense fibrous connective tissue goes to adipose tissue. There we have these Pacinian corpuscles. In the center we have the end of dendrite. It is covered with the glial capsule here, and glial capsule is covered with the outer connective tissue capsule, which contains a lot of concentrical membranes made up with the very specialized fibrocytes. So it receives pressure and vibration. Also we have Krause bulbs, which receive cold information. Also we have the branches of dendrites covered with glial cells and outer connective tissue layer. Ruffini's corpuscles, the same structure: dendrites, glial covering, and outer connective tissue covering. These three in the um, lower part of this slide are located in the organs. Here in this picture you can see which of the receptors can be located in the skin. So in the papillary layer of dermis we have Meissner's corpuscles. Here they receive touch. In the epidermis we will have free endings. And in the lower part of the skin we will have the Pacinian corpuscles. Merkel discs are located also in the skin, in the basal layer of the epidermis. This is the so-called muscle spindle. It is located inside the skeletal muscle. It serves for protection against overstretching and breaking rupture of the muscle. So it consists of several smaller in size than normal muscle fibers. This is the normal muscle fiber. This is the specialized muscle fiber of the muscle spindle. It has nuclei of myosin which can be located as the chain or as the group in the middle of the cell. Of the muscle fiber. Each muscle fiber here is innervated with the dendrites of the sensory neuron, and when all the muscle stretches, if it stretches too much, for example, it causes overstretching and overstretching of these smaller fibers, and this is sensed with the ends of dendrites. They send this information towards the spinal cord, and then in spinal cord towards motor neurons, which axons innervate these normal large muscle fibers of the muscle, and as the result they contract. So it prevents the overstretching of the muscle. If it is overstretched, it reflectory contracts. This is the function of the muscle spindle. So this is all for today. Thank you for your attention. Have a nice day.